So we spent some time in the retina talking about how information is processed there. We got that information out of the eye through the optic nerve, those axons of the retinal ganglion cells. The information that got to the lateral geniculate nucleus was then organized and summarized and sent on to the visual cortex. And so far, we've talked about the receptive fields of the visual cortex, but now let's get more into the visual cortex and how it is organized. Okay, I'm going to get a little bit of the way through, I think, this organization or mapping of the striate cortex. And I want you again to remember that with those feature detectors, what I'm talking about is the receptive field of the neurons. What are the neurons themselves responding to? And now with the organization or mapping, we're asking how are those neurons organized in the striate cortex? Well, first of all, we have a retinotopic mapping. So we really, if you remember, we saw this at the retina, obviously, because it's in the name, but we also saw this at the lateral geniculate nucleus, and we're seeing it at the visual cortex. And this really allows for an efficiency of functioning because neurons that are near each other, that have receptive fields that are near each other on the retina, are going to be near to each other on the cortex, which also means that the visual space, because you remember it's this flipped upside down image, but the visual space is, if something is close to something in the environment, it's going to be close to that. The receptive field on the retina, it's going to be close in the visual cortex, so it makes sense for putting information together as well. There is a slight displacement, but otherwise we have this retinotopic kind of mapping that gives us an efficiency of functioning. Second, we see a cortical magnification. So I have these put in this word art so that hopefully it tells you to look here. This cortical magnification is important, that we have our foveal retina. If you remember, we had that direct wiring of the cones, the kind of one-to-one wiring. Well, the fovea is taking up 0.01% of the retina. It's this really small part of the retina, but its representation in the visual cortex is more like 8 to 10% of the cortex's retinotopic map. And I'm going to show how this was determined through brain imagery and give you hopefully a better idea too of what this, remember we've done this principle of transformation, we've done this principle of representation, what this representation that we now have in our cortex, what this looks like. Okay, so we're going to look at a study by Robert Doherty and his colleagues that uses fMRI, or functional magnetic resonance imaging. And so that you know how they are getting this data. They are using magnetic fields, radio waves, and computerized enhancement. That's what an MRI uses to map out brain structures. But for the functional magnetic resonance imaging, we want to see the brain activity. We're going to measure the oxygenation of the blood in the brain, and it follows this kind of logic. This is how it works: that hemoglobin carries oxygen. It also contains a ferrous molecule. So if you've ever cut yourself and licked it, I know that sounds fun, that it has this kind of metallic taste, has this ferrous molecule. So it lines up like tiny magnets when it's in the magnetic field. In these areas where we have high brain activity, first of all, we have blood flow to that area, and we also have the hemoglobin losing some of that oxygen because neurons are using oxygen as they are working. So this causes them to respond more strongly in that magnetic field. I have this slide that has a picture of somebody getting ready to go into the MRI. And I usually ask here if anybody's had the experience, because I think it's fun to share experiences and just to stop me talking. If anybody ever wants to share their experiences with an MRI, or getting an MRI or an fMRI, please do. I know I have plans for this Monday and we don't have a lot of time together, but it is an experience. It is a loud machine that is not a consistent loudness. It has a sound to it, and then every once in a while there's this bang, 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 clank, 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 bang, 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 and there's no, it always felt really unpredictable to me. The first time I was in the MRI was for a research study, so it was a functional MRI, but then they found a small cyst on my pituitary gland, and so I have to go in and get MRIs. And actually, I find it much more comfortable to go into the MRI without the weird thing on my face that's showing me, this computer that's strapped to my face showing me the stimuli that they want me to look at. And a lot of people talk about how really uncomfortable, if you have any kind of claustrophobia, and then the noise is going on, it can be uncomfortable. I don't actually find it very uncomfortable. Sometimes the studies are putting you in a position of going, this is weird. But I usually also really recommend here that you go and do the research, but I'm not sure how research is working right now, and I assume it's mostly surveys and not these kinds of brain imaging techniques, and the more the ones that are using our technology. But if you can find one and go run an fMRI study at some point. What's happening in the fMRI, when we're measuring brain activity, we really have to measure or look at the fact that we're looking at relative activity in areas of the brain. The brain is always active. So we are detecting these changes in the magnetic response of the hemoglobin, and we have to have some kind of subtraction. I talk about this as the subtraction method, where because the brain is always active, I need to subtract out some other activity. And usually what they do is they come up with a control task or a comparison task where they think you're not using the specific mechanisms or processes that are what I'm interested in my study, but the brain is still active, and so I'm going to have you in this kind of control task, and then I'm going to have you do my experimental task, and I'm going to subtract out the brain activity from the control task from the brain activity in response to the experimental task, and say that's the activity that is important for the processes that I'm looking at. Just so you know, I think it's really important when you read in the field, and in any field related to neuroscience or looking at fMRIs, to think about that control task and think about how much you agree with that as a control task, because that's sometimes one of the real questions of what are we looking at as far as their brain activity and what processes they are doing in these tasks. I usually compare it to PET, and I've usually gone through the PET imaging and described it, but I've taken that out because he doesn't do it anymore, and this isn't, I don't think he talks about PET very much at all. If I need to, I will. But we prefer it to PET for a couple of reasons. One, there's no radioactive tracer, so people sometimes balk at being injected with a radioactive tracer, even though it's so little that it's not going to do anything. It also, but more importantly for most of what we look at, is it gives us a more accurate and detailed map than any kind of positron emission tomography. It's also less expensive. So with all of that together, the fMRIs are often the way we look at brain activity when we want to look at what's happening in a particular area or region of the brain. So Robert Doherty and colleagues in 2003 used the fMRI to determine the magnification factor in the visual cortex. And so they had participants look at a dot in the center. So it is on the fixation, it is where they're fixating, and it is then on the fovea. And then they had a stimulus light presented in two places, and they looked at the response of the visual cortex. And the two places were either near the center, so you can see that in A, in the red, it's within 5 degrees and very close to the fovea. I cannot remember now if I've said this in the class already. I think I did, but I'm going to repeat it here. When we are looking at visual angle, we have about 180 degrees of visual angle that we can see. Since our eyes are round, we do it on this kind of circle, but we're only seeing really half of the circle, right, as our eyes work. And so if we're within 5 degrees, we're pretty close to the fovea. Farther from the center, they get out past 10 degrees. That's that little area in blue. And what you can tell in the picture in A, that there's more they're using more of the visual field there in the blue when they're farther off from the fovea. And if we look at the activity and the response of the brain, what they're showing in B is the response to what's near the center. They're showing that in red, and then what's farther away, what we see the light in blue, they're showing that in blue. And you can see that even though that area that's near the center, that's near the fovea, is smaller, there's more of the visual cortex responding. And even though we have that area that's farther from the fovea, it's a bit larger, we have a smaller area of the visual cortex that's responding to that region of space. And this is a nice visualization. So if you look at where we look at it, if we're looking at, and this, I mean, getting information on our fovea is particularly important in reading. And so if we're looking at the word area, really, we're going to foveate a little bit above that, but that's okay. So we're kind of looking between periphery and area, right? What you're going to see, so smaller surface area. If you look over to the rendition, the image of that over on the right, that is the A of area upside down. You can kind of see the word surface upside down and backwards, and then above that you see the little bit of the PERI of periphery. And as we get off in the periphery, and so it gives you this idea that we have foveal magnification. And if we're looking at what's actually what we're responding to in the visual cortex, it is this upside down inverted image with this magnification of really what's important to us and what we want, the more detailed information about. So it turns out we have extra cortical space for those tasks that also require visual acuity. So when we are looking at something and getting that more detailed information, we're looking at that because that task requires visual acuity. Good acuity is associated with a number of things. 1, a sharp focus of images on the retina. 2, that small amount of convergence of the cones. And 3, a relatively large amount of cortex devoted to fovea. Okay, so my last statement here is that people with more cortical space devoted to the fovea also have better acuity. So if you remember Jessica saying those early events are really important, and what the textbook is really doing there is the importance of the early events in the visual system. So when we're at the level of the cornea lens and what's getting to the retina itself, but also that was talking about development, so what's happening during our development. And that all gets put together here in a lot of ways. It's important that we have a sharp focus of images on the retina. If you are like me and you're myopic, and especially if you started losing your eyesight before the age of 4, you didn't really have that sharp focus of images on the retina, and you're always getting new glasses to try and make that image sharper and sharper. But because that was happening when you were young enough that the visual system was still developing, you probably have less cortical space devoted to the fovea. So I usually tell this story, I'm mixing up my words, this story about my sister and I, where so we spend a lot of time walking outside. She lives on Edisto Island, and so we spend a lot of time just out in nature. I remember we were walking down the dirt road, and I said, I can't tell by the flight pattern. It looks like that bird is flying kind of like a hawk. Is that a hawk or is it a turkey vulture? Because they're far enough off that it's hard to tell one from the other. But it was kind of flying straight instead of doing that around and around the way that turkey vultures do. And she just kind of shook her head at me and said, "You can tell by the beak on that bird that it's a turkey vulture." And I just kind of felt like, I'm lucky to see a bird in the sky, don't you know? So she has really sharp vision of things that a bird was really far away. Clearly, she has more cortical space devoted to her fovea because she has really sharp visual acuity. And you can do that kind of comparison with your friends in your mind. Because again, we're here at these feature detectives. They are responding to features. They are responding to different orientations of lines in the environment and direction of movement, and then maybe how long that line is and the angle and so forth. And so then we start talking about the organization or mapping of this primary visual cortex, of the striate cortex. So how are the neurons organized? And we gave it's a retinotopic map. It has the same kind of mapping of what we see on the retina. And we have foveal magnification. So it's we're seeing there's larger area of the cortex responding to the fovea than to that peripheral information. But so far, what we're looking at is the surface of the cortex. And we also want to look below the surface. So I don't remember if I've said it in this class, but throughout our cerebral cortex, yes, because I talked about the lateral geniculate nucleus, and that has those 6 layers, and that we looked at particular columns, right? Well, we see a similar kind of 6 layers here in the cortex, and we want to look within the columns. So we want to go perpendicular to the surface, and what we see are location columns, orientation columns, and ocular dominance columns. So when they put an electrode in and it's going perpendicular to the surface of the cortex, all of the neurons within a particular location in the cortex have their receptive fields at the same location on the retina. So they're really representing a particular location in space. And I usually do this thing of standing up in front of the class and just kind of putting up a pen or my finger or something and saying, you know, if this is on your right side, then you have a particular area in the left visual cortex that is responding to this particular location. And I'm going to continue on with that example. We also have orientation columns. So neurons along one of these specific tracks also prefer stimuli with the same orientations. And what this is showing is, what the picture is showing is, well, the cortex is curved, because remember we have those gyri and sulci, and we have these curves so that we have more surface area to our cerebral cortex. And if we're going in at the surface of the cortex at A, and we put in our electrode, then everywhere along in that column, when we're measuring at A, it's going to be responding to a horizontal line. And if we look at B, so we move out a little bit at B and move along, go down in a column from the surface of the cortex, and every single neuron in that column is responding to a particular location in space and responding to a particular orientation. So it only is going to respond when, I'm going to pretend that's kind of vertical, even though it looks a little bit oblique to me, but it's going to respond to the vertical line. So here I am, I have my finger up, it's in your right visual field, it's hitting the left sides of your retinas, going back to the left lateral geniculate nucleus, going back to the cortex, and there's an area of that cortex in the left hemisphere that is responding to that location and the fact that this is a vertical line. So that particular column is where you're going to see neural firing. And the last kind of column that I'm going to talk about at all is the ocular dominance columns. So again, going perpendicular to the surface of the cortex. If we go in, we see, well, first of all, we see that 80% of neurons really respond to stimulation of both the left and right eyes, but most of our neurons respond better to one eye or the other. So we have these neurons that have an ocular dominance. And again, we're going to see these location and orientation columns, but also with a left eye dominant, right eye dominant neuron, if that makes sense. So these neurons with the same ocular dominance are also organized into columns. Discussing the visual cortex, when we started talking about those feature detectors, this is a reiteration of those simple cells, complex cells, and end-stopped cells that Hubel and Wiesel discovered. I went through and gave examples of each, so hopefully they make sense that we have particular neurons that are responding to particular orientations of lines or bars out in the environment. We have other neurons, the complex cells, that are responding to a particular orientation of line. with a particular direction of movement. And then we have end-stop cells that respond to really the length, angle, orientation, and direction of movement. And how does this look? They actually have a really neat picture. I don't know if it's still in the book, and I used to have it in my slides, and I can't seem to find it now, where they don't look like these hypercolumns. They are not these perfect cubes, okay? But they're because the cortex is curved and has those folds, they are sometimes kind of wrapped around. But nevertheless, when we go in perpendicular, we see hypercolumns. So hypercolumns that include are giving us information about a particular location. They have these orientation columns. So what you can see going from kind of right to left of, we're kind of on a z-axis is where I'm putting that. We start with a vertical and then these different obliques until we get to horizontal. So we have these different orientation columns. And if you see the R and the L there within the location column, we have a right eye dominant column and a left eye dominant column. And I'm going to ignore that we also have these spatial frequency columns in there as well that are columns that are responding to either close spatial frequencies, things that are really busy and the lines are close together, versus more spread out lower spatial frequencies. I'm going to ignore those because your book ignores those. So this is kind of the idealized column, hypercolumn, looking at it as this cubicle kind of thing, but it's really kind of wrapped around and warped with the folding of the cerebral cortex. We want to get to this question of how is an object represented in the primary visual cortex in V1 or the striate cortex. How does this electrical activity stand for or represent the object in the environment? So we have, in this example, a person is looking at a tree. We have that image hitting the retina. The photoreceptors are firing. The retinal ganglion cells are receiving their information. They're sending it on to the brain, to the lateral geniculate, to the visual cortex. We have this representation in the striate cortex that is similar to the image on the retina that, remember, has already gone through a process of transformation and representation, but now we have more space that is devoted to the fixation point or the fovea as well. And of course, it's pulled apart into these hypercolumns, where a particular hypercolumn is going to give me vertical bar, horizontal bar. What kind of feature am I looking at? Is it right eye, left eye, dominant, and so on within that? So if we're asking the question, how is an object represented in the visual cortex or in V1? Okay, so we're looking at the tree trunk, and we're seeing activity in a number of separated orientation columns. They are giving us each location, and those locations are close to each other on the retina, but these orientation columns, A, B, and C, responding to those areas of the trunk A, B, and C, are now separated in the visual cortex. So this looks quite a bit different than the stimulus itself anymore, and it becomes really clear that cortical representation is not resembling the stimulus, it's representing the stimulus. And the representation of the trunk of the tree in the visual cortex is really, again, just the first step, because we need to process this information and recognize it as the trunk of a tree, and that it's related to this bushy tree, these leaves and everything that's on the top. This information is going to go to a number of other places for higher level processing, so that we can understand what objects are in our environment and where those objects are in relation to ourselves and to other objects.